Rebuilding the body with the same level of perfection that the nature gave us is better than any other drugs, any other therapies, because the cells do the job.
Historically, medicine has relied upon developing chemical drugs to treat patients. However, the discovery of stem cells back in the 60s opened the way to a new therapeutical approach. Graziella Pellegrini and Michele de Luca are among those rare scientists who persevered with their research into epithelial stem cells until they found how to use them for medical treatments, despite the challenges involved in achieving a complex and fragile equilibrium between fundamental research, cellular biology, and translational medicine.
This is the issue, the problem of the regenerative medicine and stem cell-based therapies. It's not like pure biochemistry or pure molecular biology. And there are not many people that want to pursue this sort of path, which is difficult, you need tears, blood, sweat. It's not easy.
The two Italian scientists met in 1986 and found they were both fascinated by the incredible potential of epithelial stem cells in regenerative medicine. Driven by the same curiosity, they decided to pool their efforts and their specialities, chemistry and pharmacology for Graziella Pellegrini and medicine for Michele de Luca. They realised they could successfully apply their knowledge to cultivating epithelial stem cells in order to treat patients suffering from severe ocular and skin diseases, but soon found themselves stopped in their tracks by a nascent and complex drug regulation.
That was considering this kind of biological tissues as drugs, as standard drugs. So this meant a huge revolution in the field, because at the beginning everything was stopped. Nobody was able to classify a tissue or a cells as a drug because the regulation was not appropriate for this field. Then, after many discussions between biologists and regulators, there were some guidelines that were producing a new regulation more appropriate, more compliant with the kind of application. At this point, we started the work of certification of our therapy in order to be able to continue the clinical application, because the clinical results were very good.
We have to do several controls in order to evaluate the properties of the culture during the tissue reconstruction before the implantation of the tissue in patients. Each of these colonies are grown from a single cells, and so they have a great regenerative potential. This is really important for me to work for a real perspective in order to improve the life of the patients.
In 2015, the two Italian scientists at last obtained EU approval for the first advanced therapy medicinal product containing stem cells. This new therapy is used to treat patients with damage to the surface of the cornea caused by external factors such as chemical or thermal burns. A healthy zone of epithelium is collected and then placed in culture before being reimplanted. A procedure and medication developed here at the Centre for Regenerative Medicine in Modena, a building that meets GMP Good Manufacturing Practices norms.
Since both cell and gene therapy are advanced therapies, you need to have a GMP facility. We built up this center that contains both the basic research, which is important for the development of these therapies, and also the GMP facility, which is run by a biotech that is taking care of all the translational part of our research. It's a building where there is the coexistence of the academy, so the university, and also the biotech to bring our therapies from the bench to the patients.
We are in the same boat. We are moving with two patients, and we are moving in that same direction. I try to manage the boat. They have a big amount of science in their inside, and I try to focus the energy to the patient and to the treatment.
Alongside these activities, Graziella Pellegrini and Michele de Luca decided to move forward by further developing gene therapy.
It's quite obvious for us that the moment you know the biology of a specific stem cell and you know how to apply this into the clinics, the first thing that you want to do is to modify it genetically, because we have the genetic disease. And I personally decided to get involved in this difficult field because not only you have to control the stem cell biology, and then you have to create a large amount of skin preserving those stem cells, but you have to do all this after genetic modification.
By combining stem cells culture with gene therapy, they were able to save a child suffering from junctional epidermolysis bullosa, a skin disease which can prove fatal.
We are able to isolate from a very small skin biopsies from epidermolysis bullosa patient epithelial cell from 2-9 centimeter square skin biopsy and grow these cells and correct in these cells the genetic defects. And we are able to apply this on patients and treat this patient with this type of treatment.
Thanks to their rigorous work, which at times became tedious as science and industry needed to progress together, the two scientists have shown the way forward by providing a model for the development of new gene therapies combined with regenerative medicine.
Whatever you have learned up to now is the basis to build over something more complex, probably, so to have a more ambitious project.
There are hundreds of young people that come every day here, and I wish I could be like them again, to start again all this.